Welcome to the tissue engineering session of our symposium. You probably know that during your lifetime, tissue engineering evolved from an emerging field into mature discipline, which is affecting very many areas of medicine and our everyday life. So what we do today is building models of some very complex diseases, such as lung fibrosis or heart infarction or cancer. And we are using these models to design patient-specific therapies. We are also building models of human physiology. For example, you can study aging, and we are developing new generations of biomaterials. What I would like to emphasize is that our faculty has been at the forefront of these very remarkable advances, and they have been driving the translation of the scientific discovery into the practice. So today you will hear 4 very exciting, interesting stories from our faculty, and each of them will speak about some of their very recent research. So we will start with Professor Kei Cheng, who will speak about the use of cell-secreted extracellular vesicles to treat lung disease. And before we start, just to remind you, no questions during presentations. We will have 4 talks back to back, and then we have a discussion session, and we will be answering all your questions. Okay, welcome.
Thank you, Gordana. Thanks again, Gordana, for the wonderful introduction. So, again, my name is Kei Chen. I'm a relatively new faculty here. I moved here last July, so the lab is also expanding and looking for prospectively new students. So the lab works on cardiac and pulmonary tissue engineering using cell therapies and exosomes, which are naturally secreted vesicles. But today, for the constraints of the time, we'll just only focus on the pulmonary part. So as we know, pulmonary fibrosis is one of the deadly diseases. It's actually with 5-year survival about 30%, actually worse than many of the cancers, only better than lung cancer and pancreatic cancer. And as you can see from this HRCT image, you see this honeycomb structures. Those are the fibrotic area, and usually lung transplant is the only result. So we, as a biomedical engineer and working on regenerative medicine, the goal is to reverse or at least stop this maladaptive progressive process of fibrosis. So cell therapy or stem cell therapy is a promising option. And the field has been using cells that is not belongs to the lung, such as mesenchymal stem cells from the bone marrow or fat tissue. So our approach is try to use resident lung stem cell, but the bottleneck is very hard to grow, isolate, and kind of develop these cells. So over the last kind of 10 years, we developed this cell therapy. We name it the lung sphere cells. They are a combination of bona fide genuine epithelial stem cells, a mixture with mesenchymal stem cells, and we can do it either autologous or allogenic fashion, which means get from the same patient or from a donor, and then get a large quantity of cells in a relatively short amount of time. So showing here is our proof of concept study. Like a few years ago, we did before you can go to human, you have to treat some animal models, and this is a mouse model of lung fibrosis. So we give a chemotherapy. This is an old chemo drug that one used to treat breast cancer, and one of the side effects of those patients, they have lung fibrosis. So then we use this drug in these mice to create this lung fibrotic model. As you can see here, this is a healthy lung, which is with a lot of void space. These are space for air exchange, so this is pretty normal. And if you see this fibrotic lung, you will see a lot of this empty space has been taken up by inflammatory and fibrotic tissue. And after you inject these lung spheroid cells, lung stem cells, you would appreciate the condition not totally reversed, but much better than the diseased lung, as you can see from here as well. So that was back in 2015, it's almost 10 years ago, and then in the university. So these are the work done at University of North Carolina, Chapel Hill, and NC State University. We converted a tissue culture laboratory to a clinical grade lab that has a fair amount of GMP or quasi-GMP conversion to allow us, this is a first step, this lab is, was at NC State, and take a fair amount of regulation for the university leadership allow us to do that, because, NC State historically was not strong in human medicine. Everything we made goes to human body, the beer and ice cream, from the food department. So I always joke to our chancellor, this is the third product we're making that go to a human body. So we convert this lab, and then we get these cells manufactured at NC State and shipped to Chapel Hill for this first-in-man trial for autologous lung stem cell transplant for a patient with IPF. With Jason is our clinical PI, and this trial is ongoing. We have so far treated 12 patients and with great safety data. And we are happy we'll be reporting these efficacy data as well by the end of this year. So as we are very happy for this cell therapy, we start looking at something else called acellular agents, or the cells. When we start to looking at what is the mechanism for this cell to impart their therapeutic benefits, we get very interested in these tiny vesicles called exosomes. So exosomes are discovered decades ago, but previously viewed as garbage that cells want to get rid of, right? And then later on, we actually see literature report that exosomes are really a text message between cells, which is for cells to communicate with each other without touching each other. So you can secrete a vesicle. For example, a stem cell can secrete a nanovesicle, an exosome, and then received by an injured lung cell and tell the lung cell, hey, you have to wake up and regenerate yourself. That's one kind of text message. Another type of text message, which is not that pleasant, is a cancer cell can secrete text message, which is exosome, and received by a normal cell. And then this exosome will tell the normal cell, hey, you have to help me to evade. Right? So this is how the exosome function. And obviously, we want to utilize for the first application, which is using exosome for regenerative medicine. Now, over stem cells, exosome has advantages, which include their less tumorigenicity because they have no life. They also have more stability because cells, as a live drug, to preserve them, process them, takes quite a bit. A lot of facilities, and also they are not that stable during transportation, but exosomes are more actually stable. So as I said, exosomes, they contain a lot of cargoes, non-coding RNAs, and also they are very enriched on the surface, has a lot of binding molecules. So the field has two ways to utilize exosomes, or we call it, I'm going to interchangeably use these two terms, exosomes or extracellular vesicles, which are EVs. First of all, is using these natural EVs for regenerative medicine. And secondly, which is also a big part of my lab, is using exosome as a drug delivery vehicle. So for the regenerative medicine purpose, you heard before that I'm using these stem cells for lung fibrosis. This trial is going on. So one of my previous graduate students, Wen-Ding, she's asking the question, can we not use the cell, but only use the exosome secreted by the cells? And now one of the benefits of using exosomes is that now you can inhale, because the cells are too big for inhalation. But this exosome, they are in nano-sized kind of diameter. You cannot inhale them. So what Wen did is to put these same kind of mice with fibrosis into the chamber, and then taking the supernatant, including exosomes from these stem cells and then nebulize to this box and let the mice inhale these exosomes and secretome and see what happened. So what we see is actually very interestingly that just inhale that exosome or secretome itself, you will have this phenotype, beneficial phenotype that very much resembles what we're giving stem cells themselves. And we're taking these clinically relevant lung function tests, do these tests on these mice, and we actually see the improvement after this exosome inhalation, which is this blue bar, more close to the healthy bar as compared to the disease, the placebo control bar. So now we started with cells, lung stem cells, and now we are digging into this, like peeling the onion. We are identifying exosome as therapeutics, and also we are continuously working on the mode of action by identifying functional defined molecules, such as microRNAs or other non-coding RNAs in the exosomes, and some of the mechanisms related to anti-fibrotic pathway being modified by these inhaled exosomes. And this is actually showing this example that if you give a defined microRNA inhalation, you also have some sort of partial benefits. So the second part of my talk is 2 minutes, actually, really not second part, but seconds. So quickly go down this, how do we use exosomes for drug delivery to the lung? So this is another graduate student's amazing student, Kristen's work. So what she did is compare exosome delivery to liposome. Liposome are similar to LNP, and LNP is the carrier of the microRNA, the mRNA COVID vaccine that most of us received. So what Kristen did is she identified that in this particular inhalation lung delivery that exosomes outperform liposome in terms of deliver this both mRNA and protein to the lung. As you can see, there's stronger signal compared to the liposome. And if you use lung-derived exosome, outperform a generic cell-derived exosome. So this is actually showing some images of this. When you make the lung transparent, you can take a journey into the lung, similar to the image, in the morning, Dr. Hillman showed that we now can see these exosomes and vesicles in different parts of the lung, and also proved that they are better than liposomes as well. And Man Ray is my postdoc sitting in the audience, and now we're using this exosome. One of the applications using this extracellular vesicle to deliver a cancer immunotherapy mRNA, and this is recently published work. We see that using exosomes, you can deliver this anti-cancer drug that is outperform liposome and can extend the life of the mice with lung cancer. And we're actually actively translate this work as well. And also we're making a step further to make this a dry powder. So imagine patient now can take this home in their pocket for other indications such as cancer or cardiovascular indications. We can actually deliver this mRNA into this is actually monkey data showing that this mRNA continuously expressed in the monkey lung. So I still have 30 seconds left, actually. So I'd like to acknowledge the students in my lab and also a funding agency and my department as well. Thank you very much.
Next speaker is Kellen Liu. She'll speak about regenerative biomaterials.
Thank you. Okay. All right. So good afternoon, and welcome to the students who are visiting Columbia this weekend or this week. All right. So let's get started. So today I will focus on regenerative materials by giving you some perspective that we've gained in the past couple of years. And I will focus on two stories, and I'll get into them in a little bit. So how many people here have taken a biomaterials class? All right, awesome, great. Admitted already, no, I'm just kidding. Okay, so for most of you know that biomaterials have a long history. So the materials that, we went from inert material to biodegradable materials, now to like regenerative bioactive materials, right? So what are regenerative materials? And regenerative materials are generally made of degradable materials. And the regenerative materials are enabled by degradable polymers. Okay? And these polymers from the polyalpha hydroxyester family of polymers. And I will focus a little bit on them today. So in my lab, we work on connecting different types of tissues together in order to form complex tissues. So these connections between soft and hard tissue are found everywhere in the body. So above the neck, and the tooth, tooth are connected to bones through these ligaments. The ligaments connect bone to bone, and tendon connect muscle to bone. And in the lab, we've worked on many interfaces. Some of the active projects right now are cartilage to bone integration, ligament to bone integration for the tooth, as well as a project with Nadine Shaheen, whom you'll hear from a little bit later, on the intervertebral disc. We just got NIH funding for a new project on women's health, which is in the pelvic floor. So there are also insertions there that connects muscle to bone that holds all the organ in the pelvic floor. So how do you regenerate that insertion? So a typical project in the lab starts with someone who's interested in one of the insertions, and then we spend time characterizing it to understand, if it hasn't been characterized before, to understand what are the design criteria. And then you design for that specific application. So you can use our arsenal of biomaterials that are available or can be invented, or fabrication methods, and you will test the system in vitro and in vivo. Okay, so some of the materials that we have tested out, because there's really the biomimetic principles, using electrospinning to mimic what the cells actually make, the extracellular matrix. So they're generally nanoscale, and the method we use in this scenario are electrospinning, which some of you probably are familiar with, and you can really control the diameter, the alignment, the ultrastructure, and the macrostructure of the material, of scaffolds through this fabrication method. And so this is a story, is actually the second insertion site interface that we worked with when I started at Columbia back in 2001. The first one we worked with ligament to bone, and second one was tendon to bone for the rotator cuff. And this project was very successful through 3 PhD students, and it came through I think, 300 to 500 rats in studies, and all the way through 2 large animal trials to sheep. So we were at a point where we were actually thinking of translating this to clinic, and I was having my meeting as a team with our FDA consult, and the question they asked me was, "What solvent did you use for electrospinning?" Why are you asking me about solvent? So there's a reason why they did that, because what solvent you use makes a huge difference whether you get FDA approval in the material that you fabricate. So this led us down to sort of a path to think a little about regenerative materials as well as green processing of these materials. Just so happens that FDA actually has a very stringent requirement on what solvent you use in biomedical polymers. Okay, and you have to come from a certain family of polymers, and just so happens that electrospinning, as an old technology, has utilized non-FDA approved solvents, and so it's probably not surprising. Very few technologies are actually gone to the clinic. So we went back to the drawing board thanks to Chris Mosher, who's a PhD student who graduated actually during the pandemic. And he decided to pursue what we call green electrospinning. So how do you consider green processing methods that are actually can be facilitate FDA approval? So over here you can see that Chris checked the literature in terms of what was published back in 2021, back by 2020, in terms of the solvent that are used. Very, very few green solvents. So what are green solvents? And so we looked at the class 3 solvents in the FDA guidelines, and then we started to look at them. Can you actually use them for green electrospinning? So Chris checked a couple of standard parameters, and we compared green to traditional fabrication methods, which is primarily the TFE or DMF-driven methods. And then he was able to use a couple of classifications to identify a few solvents that worked. And I will talk a little bit about the story about acetic acid. So in here, in these following graphs, your goal is really to look at everything that should be the same, traditional versus green, okay? And as you can see here, that in synthetic polymers, we're able to achieve comparable mechanical properties. There's no change in structure or crystallinity of the material. So you could definitely switch solvents in this scenario. And cell response is very comparable. These are chondrocytes. Chris was interested in cartilage to bone integration. So he was particularly interested in chondrocyte response on these materials. And so this is when things started to become a little different. So we use acetic acid compared to traditional DMF formulations for growth factor delivery from nanofibers. And what was interesting that this solvent was much more biocompatible in terms of preserving protein structure, as well as in terms of the release kinetics. So next thing Chris did is that he was, because we're integrating with bone, so we have to consider hydroxyapatite nanoparticles, incorporation of nanoparticles into these fibers. So he also mapped that out to see that whether we saw a difference. Again, all green light there. And another student in the lab, San Wang, so these materials are very complex. So we're beginning to think about how do you incorporate AI or machine learning into material synthesis. So ultimately, you can actually minimize the number of experiments you do, but actually have predictive fabrication of materials. So we began to develop neural networks for this purpose, and these are some of the data that shows, this was a high fidelity processing in both systems. Finally, the polymer that we checked was a biological polymer, because we use synthetic polymers and synthetic nanoparticles. In the biopolymers, again, you saw actually a better response. We're able to actually create a strong material with high ductility. Okay? And this is a collagen fiber-based platform that Phil has developed. So these are examples of how, for a regenerative material, how you can actually incorporate sustainability and also green and biocompatibility considerations into fabrication of these materials. So my title mentioned a second perspective. So these are the non-degradable polymers, the non-degradable plastics. So that's the other perspective. And if you ever work with metals and ultra high molecular polyethylene, they are not degradable materials. So you have sort of a cradle to grave scenario. The material goes to the landfill, right? But degradable polymers, biomaterials that we use, regenerative materials, they're not just regenerative in function, but also regenerative in life cycle. So this is a consideration that has been began to sort of spread beyond biomedical engineering into sort of everyday materials. How do you generate materials that have a circular life cycle outside of medicine? And this is in collaboration with Thean Shiros, who's a faculty member at Fashion Institute of Technology. So we went down the path of the other regenerative materials. So this one, there are some materials from the ocean. We were actually using them to make cartilage. Okay, alginate. Some of you probably are familiar with this material. And Thean came along and said, can you make an alginate fiber that's strong enough to make clothing? Okay, so this was a very straightforward project. So we gave it to a master's student, Romar Antrobus. He was a new master's student in the lab. So he took this to a new level, and he was able to make very strong fiber. And that actually was translated into a startup. And the startup made textiles. And these are the yarns that are called alginate yarns that are made based on alginate. And then also the product was first launched on the Paris runway last year. So this is an example of a material from the ocean. And then we also went a step further in another startup, which we started putting color into fibers, because another source of pollution is color. The chemical that used to dye, so that I could wear this blue jacket, for example, create significant impact. So again, this is leveraging biology to do that. So what's next? So we spent the past 5 years looking at what are the platforms that you can actually leverage from nature. Okay, that cells can fabricate. So I show this slide here because, as you can see here, the nanofiber here are 39 to 40 nanometers. We cannot electrospin fiber at that level, but the cells make them naturally. So this is a bacteria that actually makes nanomaterials this way, and also has mechanical property comparable to what you want to use. And this is Romar, who became a PhD student, and these are the materials that he can make that are at the physiologically relevant scales. And he also was able to purify these materials for biomedical applications, showing that biocompatibility in the macrophage cultures. And finally, you can process this material post-fabrication to facilitate other functionalities. Here you can see that this material is highly flame retardant, and that's, again, you can thinking about applications beyond biology. Okay, finally, in summary, I gave you two perspectives of some of the work that we're pursuing in the lab on regenerative materials. So biomaterials are inherently regenerative for medical applications. At least those are used for tissue engineering. But you have to think about how to fabricate them in a biocompatible and sustainable manner. And then you can also think about regenerative materials beyond biology and leveraging biology to do so. And this is my final summary slide. So a lot of biomaterials focus on mimicking nature. For many, many years, that's what we did. And now we're beginning to think about how do we purpose nature. So I gave you examples of two. And then I'm looking for a student who can come in and actually improve upon nature, because I think that's really the future of biomaterials. All right, this is the most important slide for all the folks who did the work. And thank you for your attention.
Thank you, Helen. Our third speaker is Ricardo Cruz Acuna. So he will be speaking about the translational studies of cancer.
So much. Thank you, Gordana, for the introduction, and thank you to the organizing committee for inviting me to talk. I'm very excited to talk to you guys about my lab, and also one of our most recent project that was published last year in the Journal of Clinical Investigation. Our lab is a new lab. We are called the Biomaterials and Tumorigenesis Lab, and the overarching goal of our lab is to create new novel engineering platforms to study novel mechanisms of tumorigenesis. We use the knowledge that we gain in the lab and leverage that knowledge in the context of personalized medicine applications and also therapeutic targeting. We are particularly interested in studying the contributions of the extracellular matrix properties, as well as disease-relevant mutations in the transformation of pre-neoplastic lesions into cancer. And some of the models that we use in the lab are engineered hydrogels, patient-derived 3D organoids, as well as genetic engineering of human-induced pluripotent stem cells. Now, I'm going to talk about our most recent project that was published last year, which is focused on esophageal adenocarcinoma. However, the models and the concepts that we create and study in our lab can be translated to other cancers and other diseases, and that's something that we do in our lab and in other new projects that we have that are about to start. But first, the reason why I was interested in studying esophageal adenocarcinoma, it's because it is the most common esophageal cancer in the Western world, and that includes the U.S. Unfortunately, patients that are diagnosed with esophageal adenocarcinoma have less than 50% chance of survival within a 5-year span, and that survival rate drops dramatically upon disease progression. Therefore, it is of my interest to understand what makes this disease so aggressive, so we can better then treat it. Our lab, as well as other labs, have found out or have done a lot of significant studies showing that the changes in the extracellular matrix properties significantly affect the progression of cancer. In the case of esophageal adenocarcinoma, we show in patient samples that the expression of different components of the extracellular matrix, such as hyaluronic acid and fibronectin, are significantly increased in the disease state as compared to the normal counterpart. What this suggests is that there are changes in the physicochemical matrix properties that are happening as a function of disease progression. Now, the problem is that there are no current in vitro or in vivo models that can be used to dissect the contributions or the independent contributions of those biophysical versus biochemical properties in cancer. And that's what we're trying to do in the lab, establish models where we can study those contributions independently. Now, in order to create such a model in the lab, we first need a cellular component. And the cellular component that we're working on is patient-derived tumor organoids. For those that are not familiar with patient-derived tumor organoids, Basically, we create them in the lab by starting from a patient biopsy that we receive from the clinic. We process that patient biopsy into a single cell suspension, and then we culture them on a dish in 3 dimensions. And those cells are able to form organoids that resemble the histopathological features of the primary tumor that they're derived from. Something also very interesting about these organoids is that they can respond to different therapies that are available in the clinic in a differential manner. But what's more interesting about this is that the response that these organoids have in our hands to drugs correlates with the response that the patients that they're derived from had in the clinic. Therefore, not only these organoids resemble a lot of the characteristics of the primary tumor, but they also serve as avatars to the drug response of the patients in the clinic. Now, to culture these organoids in the lab, we need a biomaterial. The biomaterial that we use is an engineered hydrogel that we call NORHA. This hydrogel is a hyaluronic acid-based hydrogel that was developed in collaboration with Jason Burdick's lab. And among the only few components that this material is made of, it includes the hyaluronic acid, an adhesive peptide, and a degradable cross-linker. And I can talk about the different benefits or advantages of this material, but the one that is most relevant to this talk is the fact that we are able to control the matrix stiffness, which is a biophysical property of the gel, without affecting the biochemical properties. What that means is that we can increase the matrix stiffness when we increase the cross-linker density, but that does not affect the amount of hyaluronic acid or other cell adhesive peptides. So we can control the matrix stiffness independent from the biochemical properties, and that allows us to study how that contributes to cancer progression in our organoids. So that leads us to our goal for the project, which was to understand how stiffness modulation of the engineered hydrogel can activate esophageal adenocarcinoma-associated genes in these patient-derived organoids, revealing novel underlying mechanisms of the disease. So what's interesting is that when we encapsulate these organoids in hydrogels of different stiffnesses, we observe a significant increase in the size of the organoids as well as the number of organoids as a function of matrix stiffness. So our stiffer hydrogel that we worked with was 1,000 pascals in storage modulus, which falls within the range of the tissue stiffness and the native tissue. Hence, that the range of stiffness is significant to the model and is relevant to the disease. And what's interesting of this data set is that we're able to recapitulate in vitro what some clinicians have seen in vivo, which is that cancer cells thrive in a stiffer microenvironment. And we're able to model that in our lab. Now, the next question is, how are the cells translating those biophysical signals into intracellular signaling pathways that are causing this phenotype? So we decided to look into the YAP-SOX9 axis. Why are we looking to the YAP-SOX9 axis? Is because these are considered oncogenes, and in the case of esophageal carcinoma, the activation of the YAP-SOX9 pathway endows stem-like properties to the cancer cells. So when we looked at the expression of YAP and SOX9 at the transcriptional and at the protein level in our organoids within the different hydrogels, we observed that there's a significant increase in the expression of YAP and SOX9 in the organoids within the stiffer gel as compared to the softer gel in both transcriptional and protein level, suggesting that there's a matrix stiffness dependent effect on the activation of these oncogenes. Now, the next thing we looked into was the cell proliferation as well as CD44 expression. CD44 is a marker of cancer stem cells. And as you can see from the graph as well as from the images, there's a significant increase in the number of proliferative cells as well as CD44 expression in the organoids within the stiffer microenvironment as opposed to the softer microenvironments. What this data suggests is that there's a matrix stiffness dependent effect in the stem-like properties of these esophageal adenocarcinomas via YAP pathway. Now, given the hypothesis or what we observed, how do we translate that information into something that is relevant to the clinic and from a therapeutic standpoint? So what we thought is that if our hypothesis is that matrix stiffness controls these stem-like properties via YAP, what we propose is that if we inhibit YAP, we can revert the effect of that stiffer microenvironment in the cells. So to do that, we adapted a what we call an engineered xenograph model. So basically, we embedded organoids in hydrogels of different stiffnesses, and then we implanted those in the back of the mice as xenographed for 4 weeks. At the end of those 4 weeks, we collected those implants and we evaluated organoid size, generation, as well as some esophageal adenocarcinoma protein expressions. To our surprise, what we saw is that the organoids within the control mice within the stiffer microenvironment were significantly larger in size and also in numbers to those embedded in the softer hydrogel in the in vivo environment, similar to what we saw in vitro. But what's interesting here is that the organoids embedded in the stiffer hydrogel on the mice that were treated with a YAP inhibitor called verteporfin actually showed a significant reduction in number and size of organoids, very similar to the organoids embedded in the softer gel, regardless of treatment. Next, when we looked at the expression of YAP and SOX9, we noticed that there's a significant decrease in the expression of these proteins on the organoids embedded in the stiffer gel in the mouse treated with the YAP inhibitor as compared to the control. And again, that effect was very similar to the organoids embedded in the softer material, regardless of mice treatment. When we looked at cell proliferation as well as CD44 expression, we saw a similar trend, actually significant differences, where the cell proliferation and CD4 expression vis-a-vis stem-like properties on the cancer cells was significantly decreased in the organoids within the stiffer gel in the treated mice as compared to the control groups, and those effects were very similar to those in the organoids that were in the softer group, in the softer microenvironment. What this data suggests is, in fact, that there is a matrix stiffness dependent effect on not only the YAP and SOX9 activation, but also the stem-like properties of the cancer cells, and that we can revert that effect due to matrix stiffness via inhibition of YAP. In conclusion, I hope I convinced you that the data here shows that, suggests that the hydrogels support the organoid viability, growth, and proliferation. That the data also suggests that there's a matrix stiffness dependent effect on the activation of the YAP-SOX9 axis, which is an oncogenic pathway, and that our engineered 3D platform, in vitro and in vivo, can potentially be used to test matrix stiffness activated therapeutic targets using small molecule inhibitors. In our case, as the example, we use verteporfin to inhibit YAP. And with that, I'd like to thank my lab and a lot of our collaborators. I highlighted a lot of the people that were involved in this particular study from the agencies. And as I mentioned, we have a couple other new projects in the context of cancer in our lab that we're about to start, involving patient-derived organoids, hydrogels, as well as human-induced pluripotent stem cells. Thank you.
Our final speaker is Trina Arinze, and Trina will speak about functional biomaterials that are used for tissue regeneration. Thank you. This? I'll watch that.
Good. Okay. Well, I can start talking. It's on now. Well, welcome all the students today. And I'll just start talking about, so I'm just going to really start presenting some of the latest work in my lab here. And so I've joined Columbia about 1.5 years ago. So, and we are good? Okay. Okay, very good. Yeah. All right, and so yes, functional biomaterials for tissue regeneration. You've heard very nice talks already about tissue engineering, And so our approach in my lab, and really more so as of late, we've been focusing on this number 3, which is developing of those biomaterials and ensuring we're providing the right cues to cells. And either we are using those cells in addition to the biomaterials as a therapeutic approach, or we're using those materials by themselves. What we've learned over the years, we've learned a lot by combining stem cells and other cell types with the biomaterials. So we've now been able to design biomaterials that can be used by themselves to encourage tissue regeneration by utilizing the endogenous cells. And so we look at physical properties. We also look at chemical properties. Today I'm going to focus on some of the chemistries that we've been developing. But the physical properties like piezoelectric and other types of properties, my student Yasmin has a poster, so welcome to check out that poster later. So 3 applications that we're in, and I'm going to just quickly go through these. So one is bone regeneration, and that's really kind of the earlier work that we've been focusing on and continue to develop. And this is an important area where, again, if you have severe bone defects, you need some sort of, typically a bone graft or bone graft substitute to repair those defects. But there are limitations. Those donor materials tend to not do a great job in terms of fully repairing those tissues. So it can be spine fusions, large traumatic defects that you need some help. And so I always like to put this slide, and this is really earlier work of mine. I didn't take a traditional route of doing a postdoc after my PhD. I went and worked for a company, Osiris Therapeutics, and that founder actually recently passed away, Arnold Kaplan. But here I'm showing some of that work, and we utilize those bone marrow mesenchymal stem cells to repair these large critical-sized defects. And here we demonstrate that donor MSCs could be used without immunosuppression, and that was a major finding for pursuing those cells for wide applications. This product is actually on the market, OsteoCell. The biomaterial that I studied with it didn't make it, which is a bioceramic. That's not on the market in combination with the cells. Why is that, and why we're still going in bone repair is because that material is very brittle. So for skeletal defects, you really can't utilize it. And orthopedic surgeons said, no way, we're not putting that in there. And it stuck around for a long time. So alternative materials are useful still with bone repair, and again, those key chemistries, right, to really get those cells to regenerate tissues is important. And so we've been focusing on what you already heard a little bit about. This was as Dr. Liu, which is naturally derived materials. So going to some of these plant-derived materials, in particular this corn protein zein. Zein has been used or is currently FDA approved for pharmaceuticals. But we've been able to now demonstrate its utility as a tissue engineering scaffold. So very little work has been done in this area. But zein protein will degrade by collagenase, similar to collagen. And what's nice about this as a natural protein material is that it's relatively hydrophobic. So the amino acid sequences keep it from being soluble or readily soluble in water. So we've been able to show in some earlier work it has good cell adhesion properties, but it's really poorly understood. And now, let's say, if we're going to use these corn-derived proteins, well, how does it compare to animal proteins like gelatin and collagen, which are readily used for wound healing and other applications, and they're on the market? So we did a study here very recently. We just published in comparing head-to-head zein with gelatin. So if you notice in the growth, these growth graphs and ATP viability, they're tracking pretty much, right, lockstep with growth. So that's a good sign that the zein does support all of the growth as gelatin would. And in fact, it starts to do a little bit better as you go longer in culture. But what we found, which was very interesting, a student who recently graduated, she noticed when she seeded the cells onto the zein, the cells kind of disappeared. And I was like, what do you mean they disappeared? So they went into the scaffold. So we actually looked at their infiltration into the material. And so in comparison to gelatin, the zein right away, within 1 hour, had greater penetration depth. So that's important for tissue integration and filtration. So we wanted to see, well, why is that? And so we looked at integrin expression of the cells, and so what are they attaching to on this corn protein? Maybe it's different from the gelatin. We looked at a number of different integrins, and what we found was that alpha-4 beta-1 was the one that was expressed high within 1 hour on these materials in serum-free conditions. And so that alpha-4 beta-1 integrin attaches to the LDV, that peptide sequence. And interesting, that's found in protein and fibronectin as well as in the ZANE protein. So we think it may even have some applications there as a substitute for, potentially substitute for fibronectin. So then we've implanted it subcutaneous in these skid mice models where we put our favorite MSCs onto those materials, and we see that those MSCs do produce greater amounts of neovascularization. We also see significant increase in proliferation of those cells utilizing that corn protein. And in collaboration with more of a molecular biologist, geneticist at UT Southwestern, this was recently presented at the 2 weeks ago at Orthopedic Research Society meeting. We're using a genetic model here, knockout model, looking at ZANE from a metabolic approach because ZANE is rich in glutamine. Very high, relatively high amount of glutamine. And so we think this may have an effect, especially in vivo, on growth of cells as well as mineralization, bone turnover. And so he's got a knockout model here where you can knock out the glutaminase enzyme and that glutaminase activity, and therefore preventing metabolism of glutamine. So what we found here is that, again, in the wild type, you're getting good ingrowth and mineralization of the material, but what you would expect to see if you knocked out that ability to metabolize the glutamine, you get a loss in bone formation. So this is exciting. We're continuing to look at these types of models to understand our biomaterials, especially these naturally based amino acid containing biomaterials, more carefully. And then I'll just quickly run through these couple of other applications where we're developing another type of material. So for cartilage, and a little bit in spinal cord repair, we're looking at these sulfated polysaccharides. So these are going to be imitating the glycosaminoglycan content, so the sugar components in native cartilage tissue. And this is important for cartilage repair. And so again, in early development, there's key GAGs that are present, chondroitin 6-sulfate and the heparin sulfate. So we create mimetics of those because we could essentially use those. Why not, right? But if we can potentially derive materials, again, that may have improved functionality. In this case, for these types of GAGs, we want to sequester growth factors, maintain them at certain, for certain levels. This may be important to do. And so we've been able to create out of cellulose, which you already heard from Dr. Liu again. We've been able to derive from cellulose a sulfated cellulose material. So again, similar to what you see with those native GAGs. And I'm not pressing anything right. I don't know. Right? You've got these sulfate groups that are present in certain positions along those kind of monomer units. We're able to vary that degree of sulfation and really have a high degree of sulfation, which we found to be beneficial. So we've done a number of in vitro studies to look at their effect on chondrogenesis. And so we've been able to show that fully sulfated cellulose really does promote chondrogenesis. That's in soluble form. We've incorporated into a scaffold form. And in scaffold form, also in vitro culture, we see this expression of these markers when we promote chondrogenesis. And then what's really interesting, again, we start to look at the mechanism. As I said, that sequestration of these growth factors are important. So the TGF-beta3, which is an important growth factor for promoting chondrogenesis, is sequestered at very high levels on that fully sulfated cellulose. And if we compared it to the native GAGs, which is important, again, especially when you're putting in grant proposals, well, how does it compare, again, to your natural materials? And so it does better. You get higher amounts of the TGF-beta that's sequestered in comparison to, say, chondroitin sulfate C. And we can do some neat binding experiments and looking carefully at association rate constants, and we see, again, for that fully sulfated cellulose, that you get this high binding affinity or association. You see here, it's retained, and so that's an important finding as well that we've noted here. So it sequesters it, and then it retains it over a period of time. So for healing, that's important as this material is inserted for 4 weeks or 8 weeks or 12 weeks in these defects. We did some additional mechanisms. I'll skip over that for time. And again, we're looking at whether or not these materials will interact with receptors on the cells. And so we were able to show that they do interact with, say, the TGF-beta, the type 2 receptor and type 1 receptor, and there's turning on of that SMAD2/3 signaling pathway, as shown here. Well, again, when we've implanted these into these challenging kind of critical size defects, what we've seen is that fully sulfated cellulose does repair or accelerate repair of those cartilage defects within about 12 weeks here of implantation. All right, and then I'll just finish up here for 10 seconds maybe. So for neural applications, my lab is also doing spinal cord repair. And here we also are looking at these fully sulfated materials as well. And what we found is that they also sequester neurotrophins readily, the fully sulfated ones. And we've been able to show, we've been able to show in those defects that we're getting reasonable, again, integration as well as axon growth across those defects in combination with other cell types. In this case, we're using Schwann cells for repair. All right. So my time is up, correct? So I will finish here and thank my students that contributed to this work as well as the funding agencies. Thank you.
So I would like to invite all 4 speakers just to come to the front. We will take questions both virtually and here. If you have a question, just raise your hand and we will give you microphone.
So we can start with the virtual. Dr. Cruz Acuna, do you think that the stiffer the NOR-HA ECM, the more hypoxic environment was generated around the cancer cells?
Okay. Thank you for the question. When working with 3D patient-derived organoids, there is such a thing when they can grow too much for their own good, or they start creating a necrotic core, probably because of nutrient depletion to diffuse into the center of the organoids. For the extent of the experiments that we were doing, it was roughly 2 weeks. They usually don't get to the point where they grow so much that they start developing the necrotic core. So we do believe that the effects that we see on the organoids and the phenotype that we see is solely because of the mechanical properties of the gel and not necessarily for any problems with diffusion. We did several stainings of viability and we do see that there's viability throughout the organoids into the core, regardless of the conditions that we explore.
Hi, I also had a question for Professor Cruz Acuna. For the stiffness-related activation of YAP, did you observe like a kind of a no-go, no-go situation, or what was the resolution on like stiffness and YAP activation that you saw?
Sorry, what was the no-go thing?
Like, was it kind of like you saw like you were looking at like a very soft and very hard, and then the YAP activation based on that, or I guess like what was the gradient of how YAP activation was related to stiffness?
Oh yeah, good question. So yeah, we did. We looked into the gradient of YAP activation. We did see how it was a direct relation within YAP succinate activation, depending on the hydrogel stiffness. We explored stiffer gel, softer gel than what it showed there, and there's a point where it sort of plateaus a little bit. There's a point where it's too stiff for the cells for their own good. So YAP activation does happen within a particular range of matrix stiffness. When it's too stiff or too soft, then we see no differences, and that makes a lot of sense because just like normal tissues, diseased tissues also thrive in particular conditions. And when they're, I'm talking about conditions within the physical chemical aspect of their environment. And when they're out of that environment, they don't, they don't do well. In particular, perhaps in this case, is because the YAP-SOX9 axis is not being activated outside of this range that we studied. Yeah. Good question.
I have a question for Dr. Chang. So you were telling us about how like the use of extracellular vesicles, like you can use them to signal sort of diseased cells to wake up in a sense. And I was wondering what you think about like the long-term effect of it. So like, is that something that they would need constant exposure to those EVs? Like how long do you think it would take to like sort of revert back to a disease state, or if it would ever revert back to a disease state?
Great question. I think for EV, because they have a short half-life, so you do need repeated dosing. But I think the blessing point is they are quite safe and quite small, can be usually given in a non-invasive way, so that repeated dosing is possible. Now, by saying that, there's in another chapter of this technology is engineered exosomes that you could potentially deliver a gene therapy, and then this could be a one-shot and permanent.
Okay, thank you.
Hi, thank you guys so much for the talks. I had a question for Dr. Arinze. I was wondering about like what potential future directions or applications do you see for the Zane-based hydrogels in processes like immunomodulation that benefit from cell infiltration?
Yeah, that's a good one. That's a good suggestion there as a potential application, immunomodulation. Yeah. By looking at the way these cells now are infiltrating into this material, so that LDV sequence and that alpha 4 beta 1, so that's present on white blood cells. So we think that there is this fast infiltration of cells into these materials. So we've been primarily looking at for tissue engineering in general. We haven't really been looking yet about how it might affect immunomodulation, but we've also noticed osteoclasts, so immune cells as well, or derived immune cells. There appears to be more osteoclastic activity with these materials, and we don't know how that might impact if we start to go into larger, more clinically relevant models, how that may affect bone formation, but we wait to see.
Thank you so much.
Just a follow-up for Dr. Arinze, what results are you hoping to see from the plant-derived protein Zane?
Yeah. So like I said, our first application has been bone. We've been looking for its use for that. And so we think, again, based on, we see from in vitro studies and from our in vivo, that in combination with stem cells, we are getting this rapid neovascularization, ingrowth of cells, maintenance viability of cells. So we're very pleased for that. So we think it's very promising for bone. Could have wider applications.
Maybe I can ask a question, Helen Lu. Helen, with your green materials, do you think that you can also incorporate programmable degradation, because we are basically a problem of plastic degradation? Is it something you're...
Yeah, Gorana, you always ask the billion-dollar questions. Yes. I think there are a lot of folks who are looking at the degradation of these materials. What's interesting about the microbial cellulose that's formed by the bacteria, it's completely degradable through natural process. So the idea that if you make a sneaker from it, when you're done with it, you could bury it in your backyard and will just compost. Unlike plastic, which has to go to the landfill. But what they're actually doing right now is that when they're recycling plastic, right? So certain plastic are recyclable, and those plastic, when they're recycling, they're actually trying to introduce new chemistry so that they become naturally degradable in their second life. So that's where a lot of the focus in that area is. But the material that we generate are actually naturally degradable, so we don't need to incorporate additional functionality in that perspective.
I have a question online for Dr. Cruz Acuna. Great work. Do you think that your hydrogel can maintain the heterogeneous cell population of the tumor microenvironment?
That's a very good question. It's not easy to answer, but we hope so. We are, in some of the new projects that we have going on, we're trying to implement additional cellular components to our system, not only the patient-derived organs that are talked about, which are mostly epithelial cells, but also incorporate perhaps cancer-associated fibroblasts, as well as immune cells, to better recapitulate the tumor microenvironment in the in vitro level, as well as in vivo too. We have had some success growing cancer-associated fibroblasts together with the tumor organoids, and seeing how I've made some observations of how they can interact within our in vitro environment. So it's one of the beautiful things about our platform is that we can incorporate one cellular component at a time as a new variable, and to understand how each of those variables affect our system individually.
Hi, I'm Flynn. I have a question for you, Dr. Rinze. Have you investigated other prolamines, other like the protein classes for the from like that cereal grain groups?
Plants?
Yeah.
No, we have not proteins. So we are, we try to stay along the lines of materials that have at least been to some degree seen by the FDA. So we know the Zane has, right? Plant-derived materials to some degree, some of these proteins where you're getting them from, you also have to consider potentially allergies, right? So wheat, you can get proteins from wheat, from nuts, all kinds of, there are different varieties, but then people may have more allergies for those. So it's a consideration.
Immune response.
Yeah.
Awesome. Thanks.
Okay, one more question for Kim Chang. Kate, you showed us that actually the exosomes that you collected, the secretome, are very effective. And then you also engineer exosomes by picking, choosing some of these bioactive molecules. How good are your engineered exosomes at this time in terms of their effectiveness compared to the native ones?
You're talking about efficacy or?
Yes.
Yeah. So efficacy-wise, in some applications you will have to engineer because your mode of action molecules, active principles are not naturally exist in the exosome. So you have to put in, for example, the immunotherapy one, and also sometimes you need a targeting motif so that you have to put on the surface. The natural ones are usually good when you have a good indication, when you have a very efficient delivery at the baseline. But a lot of times for indications, and you need to hit different organs, and also for indications that the natural ones are not good enough, not sufficient. So we have to engineer them. Great question.
Thank you all for the talk. I had a question for Dr. Liu. Actually, when you were talking about the bacteria that made these extra fine nanofibers, like 39 to 40 nanometers, I was just wondering about that production process. Like, has there been any investigation on being able to produce them in an aligned way, or to affect material properties, mechanical properties, or for whatever application that may be? Just curious about that.
No, that's a great question. I think biofabrication is the next industrial revolution, right? Because what's really interesting about, just like cells that makes the tissue that, in our bodies, the cells that make, bacteria that makes these material, they can be influenced by the feedstock, right? Whether you use a waste feedstock, whether you co-culture them with yeast, or what kind of pH do you do them at, do you shake them? So there's a lot of ways that you can actually control. So Romar's PhD thesis actually focused on developing the whole platform based on the feed and based on post-processing. What we've also seen that in the literature that you can actually direct the migration of these bacteria using electric field, so that you can actually make a line system. So there's I think there's a lot of potential to explore embedded functionality, so that you can actually improve upon nature. Yeah.
That's so cool.
Great question.
And we have one more virtual question for Dr. Arinze. I'm curious about the degradation of the sulfated celluloses. You saw tissue regeneration, and so I assume in cell in growth. Did you see the material actually degrade, and how so?
Yeah, so over that course, by 12 weeks, the material was gone. So that sulfated cellulose is derived from cellulose, but it's no longer, doesn't behave like cellulose, so it's soluble. So that it gets cleared in the body. So the whole thing was degraded by 12 weeks.
Okay, so this brings us to the end of our session. I would like to thank all four speakers. These talks were super interesting and very enjoyable. And I would like to thank you for very, very insightful questions. So thanks everyone, and then I guess we switch to, we have a break, and then like in 10 minutes, session 4 will be starting.